Alright, so today we're going to talk about the other process of cell division, and the name of this process, as some of you might already know, is meiosis. So in meiosis, it's all about getting some genetic variation, which, remember, is the important part of sexual reproduction, and this is the central process within sexual reproduction. Just important with this meme here, where it says meiosis, you can see two cats dividing. We actually very clearly don't have meiosis here. We actually have meiosis, and like meiosis, and it's very essential that whenever making science memes, you are scientifically correct. Alright, pause the video here and have a crack at these 3 good questions, and we'll look at them together in class. Alright, so in the study design, you guys are doing Unit 2, Area Study 1 still, so sexual reproduction. We want to talk about the key events in this process called meiosis, or meiosis, that result in the production of what we call gametes, which are the sex cells. Okay. From somatic cells, including the significance of crossing over of chromatids between homologous chromosomes in prophase 1 and the non-dividing of the centromere in metaphase 1. Alright, so meiosis, what is it? Okay, it's simplified. It's the process by which we get a diploid cell, which we call a 2N cell, dividing to become haploid cells, which are called N cells in humans, okay? So that after fertilization, the offspring is once again a diploid individual, okay? What does this mean? So on the surface, this really looks like 2 rounds of mitosis, but we're going to get into the nitty-gritty because that's really important stuff in understanding why meiosis is probably one of the most important cellular processes that we have, okay? Because in meiosis, we have mum and dad here, who are diploid, making gametes, which are the egg and the sperm, which fertilize together to get a zygote, which eventually becomes baby, okay? Why is meiosis so important? It's because if meiosis didn't happen, we simply had mitosis to produce these guys, we would end up having a baby. If the first time around, we would have a baby with 92 chromosomes, and then the next time around, we would have a baby with 184 chromosomes, and so on and so on and so on. And you can see that quite quickly we're going to get to humans with a ridiculous amount of chromosomes, and that's just a nice case for horrible mutations. Alright, so, but meiosis really, it's not that simple, okay? But really the complications are in the small differences between meiosis and mitosis. So meiosis in humans uses what we called, again, 1 diploid 2N cell to produce 4 haploid N cells, okay? So we have this 1 diploid 2N cell at the start, but 4 haploid N cells at the end. But also during meiosis, the sections of chromosomes can actually switch from one member of a homologous pair to another, resulting in what we call unique combinations, and we call this crossing over. And also in meiosis, we have each gamete that's produced only has 1 version of each pair of chromosomes. And this division of the chromosomes, the pair of chromosomes, is actually a really precise process, but also it occurs very independently for each homologous pair. Lots of information here. Let's actually break down each of these little components of meiosis. Okay, the first component was that meiosis uses 1 diploid 2N cell to produce 4 haploid N cells, and that's really easy to see. All right, here we have the process of mitosis, which you guys know like the back of your hand. All right, here in mitosis we have a 2N mother cell. All right, and that 2N mother cell is producing 2N daughter cells. Okay, in meiosis, we also call them mother cells and daughter cells. So we have an initial 2N mother cell, but in this case, we're producing 4 1N, or just N, daughter cells. Okay, to do this, we actually have 2 rounds of cell division. We have meiosis 1 and we have meiosis 2. Okay, and the breakdown of it within each of these processes, they make it nice and easy. We've got prophase 1, prophase 2, and then all that kind of stuff. We have to be familiar with some really key terms to do with meiosis, and especially to do with understanding chromosomes. Okay, so here are some definitions for cell division. We have chromatin, which is the mass of DNA and protein that really makes up a chromosome. Chromosomes are pretty much just chromatin, really, really tightly wound and wound and wound. All right, chromosome, it says, like we said, tightly coiled mass of DNA. The centromere is just this middle portion of a chromosome, and what we call a chromatid or a sister chromatid, okay, is just one leg of this centromere. So here we have one sister chromatid attached to the other sister chromatid, okay? Oh, actually, go back for a second. It's really important to know that these two sister chromatids, they are literally genetically identical. They're just the result of the synthesis phase during interphase. So we also have to know what we call homologous pairs, okay? And what a homologous pair is every single cell in your body that is a somatic cell actually has two copies of each of the chromosomes. So we could say that these are the homologous pairs for the chromosome number 1. Okay, or number 2, or number 3, or number 23. Doesn't matter which one we're talking about. These are just going to be the homologous pairs. And what that means is, for a homologous pair of chromosomes, we have specific genes at the same specific location. Okay, so for example, the eye gene, so eye color, is going to be at the exact same spot on each homologous pair. The hair gene is also going to be on the exact same spot for each homologous pair. Okay, and also the location of the centromere is also going to be at the exact same spot. So a homologous pair of chromosome, we can have them as single-stranded chromosomes, like we've got here and we've got here. But after the synthesis phase, we get a replication of these chromosomes, and we still have homologous pairs. So this homologous pair came from this paternal chromosome, whilst this chromosome, this duplicated chromosome, came from this paternal chromosome, and this duplicated chromosome came from this maternal chromosome. And just like these two are homologous pairs, these two are still homologous pairs, okay? And that's a really important word to understand, okay? These might be the two homologous pairs of chromosome number 3, okay, or number 5. Doesn't matter which one it are. All right, let's go through the phases of meiosis. This is going to be really similar to mitosis. So before meiosis, we have 23 pairs of homologous chromosomes. So if we add it up, we have 46 chromosomes altogether, but there are 23 pairs, okay? We've had interphase happening, okay? So we've had already the synthesis of the chromosomes, so they're all double-stranded like this. They're all duplicated. The homologous chromosomes will actually pair up. So here we might have the two versions of chromosome number 2, all right? And then here we might have two versions of chromosome number 1. So the homologous pairs are now paired up inside the cell, and they also condense, okay? So they become thicker. And also the nuclear membrane dissipates and the centrioles appear, okay? These are these guys again. Another thing happens as well, which you might be notice happening over here. And that's called crossing over and recombination of chromosomes. Let's watch this quick animation before we talk about it.
Crossing over occurs during prophase 1 of meiosis, when homologous chromosomes align together. Once the chromosomes have paired together, breaks will occur in non-sister chromatids at identical sites. The non-sister chromatids exchange broken segments, which are then sealed together by specific enzymes. Crossing over breaks up old combinations of alleles and puts new ones together in pairs of homologous chromosomes. This process increases genetic variability.
All right, apologies for the video from the 1700s, but so pretty much crossing over is a process that often happens during prophase 1. The chromatids on the two separate homologous chromosomes. So not sister chromatids, but the chromatids on two separate homologous chromosomes. They literally cross over, okay? And when they cross over, they break off and actually share genetic information. They swap genetic information. Why is this important? Remember, you get your homologous chromosomes, one from dad and one from mum. Okay, so they don't have the same genes on each one. The sister chromatids have the same genes, but between two homologous chromosomes, you don't have the exact same, for example, eye color gene. You might have brown eyes on one and blue eyes on the other. So by crossing over, we get what's called recombination, which is this process here, where although we have still your dad's version of chromosome on one side and your mum's on the other side, we have little bits of your mum on your dad's version and little bits of your dad's on your mum version. All right, and this is going to be extremely important in terms of genetic variation. Skip to metaphase. So in metaphase 1, there's a small difference between metaphase 1 in meiosis and mitosis, and the difference here is that the homologous pairs of chromosomes now are going to line up in the midline of the cell. Okay, each chromosome is attached to a spindle fiber, right? And it's really important because we're not actually holding each side of the chromosome. If you remember, in mitosis, we sort of have, if that's the chromosome and that's the spindle, all right? So we're going to pull this way and break at the centromere right here in the middle. In my metaphase 1, we're not going to have this happen, okay? Another important fact of metaphase 1 is we have what's called independent assortment, right? And what this means is that if this is the homologous chromosome 1 and this is the homologous chromosome 2, how these sort each other out is independent to how these two sort each other out. So we could have literally have done this exact same assortment of metaphase 1, but swapped these two guys. And that's going to be really important because in this version, we're getting maybe one from dad and one from mum, but we could have just as easily had gotten both versions of our dad's chromosomes. All right, anaphase 1 is pretty simple. The spindle fibers are going to shorten, all right, and it's going to actually separate the two homologous pairs going to the poles, okay? Telophase 1, we're just going to have, once again, nuclear membranes reforming, okay? They're just going to reform around these chromosomes, and this results in two new N daughter cells, okay? And we start calling it N because although the DNA itself, the chromosomes themselves are still duplicated, okay, we actually have now only one version of each chromosome. Okay. All right. Meiosis 2 is pretty much mitosis. Okay. So in tiny technicalities, it's not. But in terms of year 11 biology, you can call meiosis 2 mitosis. Well, you can't call it that, but you can know that it's the exact same process, okay? Prophase 1, the chromosomes are all just going to attach to the spindle fibers. Prophase 2, metaphase 2, they're going to line up. Anaphase 2, the chromosomes are actually going to separate at the centromere. And then telophase 2, we're going to get nuclear membranes reforming. And then we have cytokinesis, which isn't added here, cytokinesis. Gosh, I need a new pen. Okay, and that's the cells are going to spread up. Okay, these are meiosis recap. I'm not going to go through the whole thing again. This might be a little slide that you might want to print out nice and big and keep somewhere so you can follow along with it. All right. Another thing you have to know in terms of meiosis is actually the amount of DNA that is present in each cell. At different stages of meiosis, okay? Let's label the axes first here. So this is the stage of meiosis, and this here is actually just an arbitrary value that's referring to the amount of DNA. So So it's referring to the amount of DNA. Sorry, that's horrible writing. Okay, so at early interphase, okay, so this is just, for example, at G1, what we're going to have happening is just the cell's going to work normally, okay, and we're going to have a 2N cell with non-replicated chromosomes. So we're just going to have a standard amount. We're going to call this the 1 amount of DNA, okay? What happens between early interphase is prophase 1 is the S phase. Remember the synthesis phase? So the chromosomes are all going to duplicate. So at prophase 1, we're actually going to have still a 2N cell, still a diploid cell, but double the amount of DNA, okay? Because of that synthesis phase. Then at anaphase 1, we've now actually split up our DNA, so we're going to left with 2 poles of the cell, okay? And so we're essentially having two cells, but each of those cells are haploid, okay? So we're going to call them N cells, okay? And each of those cells have half the DNA of the initial prophase cell, but the same amount of DNA as a normal mother cell. But even though it has the same amount of DNA, it only has one version of each homologous chromosome. Between anaphase and prophase, nothing's really going to happen. We're just going to have the finalizing of pretty much 2 daughter cells that are N or haploid each, okay? But once we go through meiosis 2, we're going to be left with cells that are not only haploid, but now, not only are they haploid, but now they actually have half the amount of DNA as the original cell, okay? So here we have one mother cell, all right? Here we have still one mother cell, okay? Here now we're during this phase, we're sort of getting to 2 daughter cells. But finally, at the end of meiosis, we're going to have 4 daughter cells. We're going to have 4 daughter cells. Okay? All right, so where is all the genetic variation? Okay? Each gamete here is unique. 1, because the movement of chromosomes at anaphase is independent, all right? So we call that independent assortment. This little diagram here is really handy in showing that because it shows this original mother cell, and this original mother cell in anaphase can either sort its chromosomes like this, all nice and aligned, getting us 4 daughter cells that look like this, okay, so like those ones, or it can align its chromosomes like this, so sort of in a staggered way, and we're going to get 4 daughter cells with chromosomes that look more like the ones on the right here. But we also have crossing over, which occurs during prophase 1, okay? And this is also a massive component to why we get so much genetic variation. How often does crossing over occur? Pretty much every time we have meiosis, at least one of your chromosomes are going to do a bit of crossing over. How diverse? Well, let's look at a bit of maths, okay? Let's make things easier for us and just have 2 parents with each having only 4 chromosomes, so 2 homologous pairs. By the way, we call this a 2N equal 4. Okay, so each one of these parents are 2N equals 4. Humans, for example, are 2N equals 46 because we have 23 pairs of chromosomes. So if we ignore crossing over, the genotype of the offspring of these 2 parents could actually be one of 16 possibilities, right? So the female can produce eggs that may be A1, B1, so A1, B1, or it could be A1, B2, or it could be A2, B1, or A2, B2. Okay, the male, on the other hand, can produce A3, B3, A3, B4, and so on and so on. Okay, so pretty much just a mixture of these. If each one of these gametes, we could call them, can join with one of the others, so A1, B1 with A3, B3, for example, and we actually get a total of 16 possibilities here for a diploid 2n number of 4, okay, so that 2n equals 4. How we work that out is simply that we just use this form of 2 to the power of n, where n is the diploid number. So if n is the diploid number in this one, 2n equals 4. So 2 to the power of 4 gives us 16. All right. Now, remember that humans actually have a diploid number, 2n, which equals 46. So 2 to the power of 46 equals approximately 7.04 times 10 to the power of 13, which is like, well, it's literally about 7 with 13 zeros, all right? So which is a number that I can't actually say. But what if we add crossing over? Remember, crossing over is a thing. So once crossing over has worked its magic, okay, the chances of actually having the same genetics as another individual, even if you have the same parents, even with a small diploid number such as the 4n, 2n equals 4 that we just worked with, are essentially infinite, okay? So you're never going to have siblings that look like you, even though here in my case, my siblings sort of do look like me. They're not exactly the same as me. They're just inferior versions.